Australian pied budgerigar mutation. The Australian pied budgerigar mutation is one of approximately 30 mutations affecting the color of budgerigars. It is the underlying mutation of the banded pied variety. Appearance. All pied budgerigars are characterized by having irregular patches of completely clear feathers appearing anywhere in the body, head, or wings. These clear feathers are pure white in blue series birds and yellow in birds of the green series. Such patches are completely devoid of black melanin pigment. The remainder of the body is colored normally. The Australian pieds are very similar in appearance to the clear flight pieds, with a nape spot, clear areas on the wings, and a clear area on the breast. They differ from the clear flight pied in the nape spot, which is not always present, and in their feet, which are usually pink. But the main point of difference is in the clear area of the body, which in the Australian pied is located in the middle or lower breast, with the upper breast being always normally colored, so that there is a clear division between the mask and the breast, just as in normal birds. In the clear flight pied, the clear area on the breast, if present, is almost always adjacent to and running into the mask. Australian pieds often have larger clear areas on the wings than clear flight pieds, with all primaries and many secondaries often clear, but this cannot be taken as a distinguishing feature, as the pied areas are very variable in extent in both mutations. Australian pieds have the usual white iris ring when adult, distinguishing them clearly from recessive pieds, which have no iris ring at any age. In the Australian pied, the clear area often takes the form of a band running across the breast. Birds with such bands are highly prized, especially if the band is clear, sharp, and symmetrical, and the feature is now quite common and distinctive due to selective breeding. Such birds comprise the banded pied variety. Historical notes. Many isolated appearances of pied or variegated budgerigars were reported in Britain, in continental Europe, and in Australia in the late 1920s and early 1930s, but reliable reports of breeding results and detailed descriptions of their appearance during that period are rare. One of the earliest reports of the appearance of a budgerigar which could have been an Australian pied was of a bird owned by W.G. Bowden. It had a clear nape spot, and its breeding behavior clearly showed a dominant inheritance pattern. Mr. Bowden obtained or possibly bred the bird in 1930, when he did not report its source. The bird, a cock, was basically a light green, but it had a yellow patch on the back of the head, another on the base of the rump, and a yellow streak, about 1/4 of an inch in width, from the left wing butt to halfway across the breasts. A number of its flights were reported to be white or yellow. When mated to an unrelated hen in 1933, this cock produced 14 young over 3 nests, of which 5 showed some clear feathers on the nape of the neck. This could have been the first report of a clear flight pied or an Australian pied. Several similar pied birds were reported around the same time in Germany, bred by Herr Crab and separately by Herr Schuck, by Madame Lequier in France, by G. Wilson, and T.L.S. Dooley in England, in Holland, and in Scotland. But detailed descriptions and the mode of inheritance are unknown. The present-day Australian pieds, including the banded pied variety, are believed to be descended from a strain first established in Sydney in 1935 by Keith Ings. They were first imported to Britain in 1957/8, when Mr. M. Cooper of Caerleon, South Wales, bought 2 such birds, a pied green and a pied grey, both cocks, from a dealer in Bristol. Most of the Australian pieds in Britain are descended from the Cooper strain. Genetics. The Australian pied allele is dominant over its wild-type allele, although with less than 100% penetrance. The extent and distribution of the clear areas shown by both single-factor SF and double-factor DF Australian pieds are variable. The range of variability of the 2 genotypes appears to be identical, so it is not possible to determine the genetic makeup by considering the extent of the clear areas. In both single and double-factor birds, this variability ranges from no clear feathers at all, via just 1 or 2 clear feathers, to over half the body area affected, although the clear areas in cocks tend to be larger than those of hens. The Australian pied gene is located on one of the autosomal chromosomes. There is no known linkage of this gene with any other mutation. There is no universally accepted genetic symbol for either the locus or mutant allele, so the symbol PAL+ for pied Australian will be adopted here for the wild-type allele at this locus, and the symbol PA for the Australian pied mutant allele. The factors governing the extent and distribution of the residual pigmentation are not known, although it is likely that at least some factors are sex-linked due to the different ranges in variability of the sexes. Unlike the clear-flight pied, the Australian pied does not produce any dark-eyed clears when crossed with the recessive pied.